Hi everyone, we're looking at chapter 3. This is all about biological molecules, so different types of things that you might encounter in your studies in biology. Your cells are made up of all sorts of these types of molecules, which we're about to talk about. Basically, there's 4 classes into which you can pretty much classify all the different types of things that are inside your body and inside your cells. And all of those things are either going to be carbohydrates, proteins, lipids, or nucleic acids. There you've got it. 1, 2, 3, 4 major types of molecules. Now, these things can all be what are called polymers, meaning they are really super big and they're made up of lots of repeating units of what's known as a monomer. Okay, so polymer, the word poly means many, mono means one. So if this was kind of an example of a macromolecule, a big molecule with a bunch of repeating parts to it that just kind of went on and on and on, each one of these individual parts would be called a monomer, and the whole thing together, which is made up of many monomers linked together, would be called a polymer. Does that make sense? It'll be important to kind of keep in mind, because one of the things that we're going to talk about is the names of the polymers for each of these different types of classes of molecules, but then we also are interested what are the individual pieces that we link together to make up these big, long, big, huge polymers. So the place that we will start is going to be the process by which we can either make something a polymer, or there's another process which can actually break apart polymers to back into the individual monomers. So you can either build up or break down a polymer, and the 2 processes that are responsible for this are dehydration synthesis and hydrolysis. Dehydration synthesis is the one we have here, and it's got some curious naming. You'll see that it says dehydration, which means to lose water, right? And then the word synthesis literally means to build. So you would expect that this is probably the process that's responsible for linking up monomers to make a big, long polymer of some kind. And for an example, I'll show you 2 molecules. These happen to be sugar monomers. Okay, so each one of these is an individual unit from which the big molecule is made, so it's called a monomer. And in particular, the one that we're seeing here is of sugars, so these are carbohydrates. But in either case, what usually happens in dehydration synthesis is that the hydroxyl group of one monomer and the hydroxyl group of another monomer will join up, and the OH and the H will form water, which will then leave, and then we end up with this scenario here. So where before we just had 2 individual monomers, now we have one linked set of the monomers. So now it's getting, we had 2, and now we just have 1, and they're bound together here. And in the process, water was extracted from the 2 monomers. So that's why we call it dehydration, because when you pull water out, that's the result of these, the formation of, or the binding of these 2 hydroxyl groups. So that's what builds, this is the responsible for building big, long polymers, and if I wanted to add another one, I could, and then add another one, I could. And so this whole process, dehydration synthesis, can happen over and over and over again to build super big molecules. On the flip side of that, we have another process that's responsible for breaking stuff down, and that's hydrolysis. The word here literally means water breaking. Lysis means to break. Hydro means water. So this is just the opposite. Let's say we have a series of molecules that are bound together. This example just shows 2, but in reality this could be a whole bunch of molecules attached to one another. Well, if you add a little bit of water to that, the water can actually go into this bond and break it apart into the original 2 units. So hydrolysis breaks down polymers, and dehydration synthesis builds polymers. Okay, if you can remember that, that's good. All right, moving on then, there's a little animation here, which you're welcome to go view if you like. The first class we're going to look at then are the carbohydrates. And the carbohydrates have a common structure. Basically, it's a bunch of carbons linked together, and each one of these is going to have an H and an OH. Okay, so that's why we sometimes refer to carbohydrates in this fashion. For every carbon, there's going to be 2 hydrogens and 1 oxygen. Okay, and the number of carbons there are can vary. Oftentimes it's 6. Sometimes it can be 5. Sometimes it can even be 4 or 3 or more, or even more than 6. But 5 and 6 carbon sugars are probably the most common. Okay, so each one of these has a H and an OH on it, and then usually the last one is going to have a group that looks like this, and then this one would have a H, OH, and H, something like that. Okay, so these ones in the middle, they have that common theme, H and OH, but then the ones on the either side of the sugar might be a little bit different. This one has what's called an aldehyde group. It's kind of like a carboxylic group, except instead of an O here, there is no O, it's just an H, so we call it an aldehyde group. And then here we just have the same OH and H, and then there's an extra little spot, so another H binds onto there. Okay. The shape of these, they can either be linear or they can be in a ring form. So this is a picture showing a linear form. Okay. Keep in mind when you're looking at one of these types of pictures, the lines refer to a covalent bond. Also, the carbons, for whatever reason, the way that these molecules are written, or the way that they're shown here, the carbon is not identified, it's just assumed. And that's generally the case when you have big organic molecules. Anywhere you see a point, it's assumed there's a carbon there, even though it doesn't say a C there. Assume there is a C here. Okay, but in either case, this would be one sugar that's in a linear form, and you can see each carbon has got the H and the OH, OH, H, H, OH. You get the picture. And what can happen is the aldehyde group on one end can combine with a hydroxyl on the other end to form a ring. Okay, and so basically what happens then is that you have oxygen that's kind of tying together the two, the head and the tail of the linear part of the molecule, and so now it's in a ring form, and these can convert in between. This would be an example of a 6-carbon sugar, but sometimes you could have a 5-carbon sugar, such as this one. You can see 1, 2, 3, 4, and then 5. There's another carbon sticking out there. Okay. So this is what sugars look like, and you can have a whole bunch of different types of them. The first are the monosaccharides. This word saccharide literally means sugar, and then mono means one. So this is just things that are molecules that are one sugar. And a couple key examples here is glucose, galactose, and fructose. Glucose, I'm going to talk a whole bunch about glucose because this is the stuff that you and I primarily use for making energy quick and also storing energy quick. So if you had some breakfast, in the morning, within a couple of hours, your liver is already breaking down that food and taking out all of the glucose molecules and linking them together to store them for later use of energy. Okay, and that is kind of if we go back and talk a little bit here, that is kind of one of the main purposes of carbohydrates. Not exclusively, but a main reason why what we use them for is quick energy. You can store energy quickly by attaching sugars together, or you can make some energy quickly by breaking down sugars. All right, so that's glucose, and glucose is the really common one. We'll see it time and time again. Galactose and fructose are two other examples. Here's a diagram showing these 3 different types, and you can see they're going to be pretty similar, right? They all have the same formula, C6H12O6, and that goes along with what we said earlier, that there's usually a 1 to 2 1 ratio of carbon, hydrogen, oxygen, and we see that here, 6-12-6. And so these are 6 carbon sugars. We call these hexose sugar. If it was a 5 carbon sugar, we'd call it a pentose sugar. And for example, the only difference that you see between glucose and galactose is one small little difference. Can you spot it? It's right here. This thing and this thing. On this carbon, so this would be the 1, 2, 3, 4th carbon down. The OH is on the right side, but on this one it's over on the left side. Okay, they have the exact same formula, it's just a little bit of a different shape. When that happens, it's called an isomer. So isomers are molecules that have the same number of atoms, so the same molecular formula, it's just the shape or the arrangement of them is a little bit different. So these are isomers of one another. Fructose is still the same formula, but it's got a little bit more of a more noticeable difference. You can see that the double bond to oxygen is on the 2nd carbon versus the 1st carbon. Okay, in either case, that's some examples of what a monosaccharide would look like. I have a quick review question here for you. Taking a look at the structure of these sugars, would you say that they're hydrophilic or hydrophobic? Take just a quick moment to see if you can think about that for a sec. The answer here is that they're going to be, well, let's take a look. We know that there's an oxygen here, right? So that's going to be a slightly polar part of the molecule because oxygen is highly electronegative. And then we also have a whole bunch of interactions here with oxygen and hydrogen, and we know oxygen doesn't share fairly with hydrogen, so this would be a slight negative on the oxygen, and then that would leave the hydrogen with a slight positive charge, and that's going to be the case anywhere you see one of these. So there's lots of little spots in the molecule where there's going to be these slight electrical poles where you have a slightly negative and a slightly positive charge difference. So that means it's polar, and polar things are hydrophilic. So that's why sugars generally will dissolve in water, and that is going to be the case for like table sugar. When you put that in water and stir it up, it dissolves. Okay, good. The next one down then are the disaccharides. And by just kind of looking at the name, you can probably deduce that these are compounds that have 2 sugars. Okay, so car means sugar, di means 2, so 2 sugar compounds. And what how these are formed is if you take 2 of the monomers, okay, so 2 of the monosaccharides, which in the case of carbohydrates, the monosaccharides are the monomers that make up the polymers, okay. So in this example, let's say if we took a monomer of glucose and a monomer of fructose, or a monosaccharide of glucose and a monosaccharide of fructose, and then the hydroxyl groups on these two, they kind of combined and released water in the process, and then made a molecule that was 2 sugars linked together. This is called a disaccharide. And an example of this is sucrose. Sucrose is table sugar. So this is the stuff that you buy at the store and you bake cookies with and you put in your tea and that type of stuff. A quick question here for you. Do you think that the formation of this bond, which is called a glycosidic bond, that's the bond that attaches 2 monosaccharides together, do you think the formation of that bond was made through hydrolysis or dehydration synthesis? See if you can think about that for a second. The answer there is that it's going to be made by dehydration synthesis. Remember, this is going to build, and this is going to break. So these combine to form water, and then water is going to leave. We don't see that in this picture per se, but that would be what was happening. And in the process, we've linked 2 things together. They're bound by a glycosidic bond. All right, more examples of disaccharides. So we said sucrose is the one that we just mentioned. That's table sugar. Another type of disaccharide out there is maltose. That's the type of sugar that would be found in grains like wheat. And then lactose is another one. This is the type of sugar that's found in milk. And some of us actually do not have an enzyme which can break lactose that comes from cows or goats. And so if that is the case, that's usually referred to as being lactose intolerant. And then some of us, though, we do have a little bit better ability to break down that 2-saccharide sugar, lactose. All right, you can also have a polysaccharide. Poly means many. And again, this word saccharide means sugar. So these are the polymers, okay? These are the really long sugars made up of many monosaccharides linked together. And a couple examples of these, we have starch, glycogen, cellulose, and chitin. The first two are going to be more energy storage molecules. The last two here are going to be more structural molecules. The starch is going to be one that's found in plants. Glycogen is going to be one that's found in animals. And then cellulose is one that's found in plants. Chitin is one that's found in animals. And we'll talk a little bit at greater length at each of these. So we'll start with the starch. Again, this is found in plants. The varieties of starch that are out there are amylose and amylopectin. There's probably a whole bunch of other varieties, but these are some of the ones that are most often talked about. And so if you bite into a potato, you know, if you're eating your French fries down in the cafe, the French fries are full of this stuff called amylopectin or amylose starch. Okay, so this is when plants make sugar by converting sunlight and water into sugar through photosynthesis. The way that they store that is a big, super long polymer of carbohydrate. So what you're seeing here is each one of these little things, this is a molecule of glucose. Amylose and amylopectin are made from linking up tons and tons and tons of these little glucose sugars. Okay, and how it works is when energy or when sugars are being made, they're linked together like this through dehydration synthesis. But let's say the sun stops shining, so they can't make sugar anymore. What they're actually going to do is then start to reclaim some of this. They're going to break these apart and then break down each and every one of these little sugars to make some energy. Okay, so that's how they store and release energy. Again, but it's called starch when we're talking about plants. Amylose is the long chain. Amylopectin is the same thing. Each one of these are made out of glucose. It's just that it's branched. You can see we've got these little branches coming off. If you're curious to know what it looks like, I'm not too particular that you know this figure here, but just if you're interested, you can see we've got these glycosidic bonds that are forming between each one of these little rings of which is a glucose molecule. And sometimes we call this a 1,4 bond because it's a bond between the number 1 carbon and the number 4 carbon of neighboring sugar molecules. And when it's branched like this, sometimes we call it a 1,6 bond because it's between the first carbon and the 6th carbon. Okay, so usually if you have a branching molecule, it's going to be a glycosidic bond that's different than the linear form. All right, glycogen is very similar to starch. It's just that it's the one that's found primarily in animals. And it's characterized by being super branched. Okay, this stuff is highly branched. And you can see, I mean, there's a lot of different branches coming off of branches. But despite that, it's very similar to starch in the sense that each one of these things is made out of glucose. And so, for example, when I eat my cereal in the morning, my liver, glycogen, this stuff is usually made, it's made in the liver, and it can be kind of stored in the muscles and in the liver as well. Okay, but let's say I'm eating my cereal, and in my digestive system I'm going to break down all of those complex sugars and things, and then my liver is going to reassemble them into this big storage molecule called glycogen. So my liver is continually taking all of the sugars that have been absorbed into my bloodstream from my small intestine, and then it's linking those together to form these big chains of glycogen. And this is in my liver, and it also gets made and stored in my muscles. And then when I go throughout the day, my liver can also break these molecules down and release them so that I have kind of a steady stream of energy that can be produced throughout the day, even though it's going to be a few hours until I eat next. And glycogen is going to be the primary thing that muscles will use when they start working. So if you were to get up and start walking around your house or your apartment or whatnot, the first energy substance that your muscles will use is this glycogen. They're going to start immediately breaking these big molecules and taking stripping the individual glucose monomers and making energy out of them. Cellulose then is another one that's found in plants, but this one is not so much used for energy. This one is more used for structure. So cellulose looks like this. It's a little bit of a different way that it's made out of glucose. So each one of these things are glucose, but rather than in the case of starch or glycogen, the bond between each glucose molecule is a little bit different. So I don't know if you caught that, but if I go back to like a molecule like this, starch, you can see that each one of these bonds is like this. Okay, the O is pointing downward. Well, when I come over here, they alternate. One goes down, one goes up, one goes down, one goes up. And because of that, it's kind of a curious thing. We don't actually have an enzyme that can get in here and break these down. We don't have a substance in our body that can get in there and break these apart. So it's very hard for us to digest cellulose, which is good actually for us because cellulose ends up being fiber. This is the stuff that passes through your small intestine and your colon and kind of cleans you out, and it gives you a little bit of roughage to kind of it's almost, think about it being like when you eat like a lot of salad or something like that. It's almost like a brush that cleans out your digestive tract. So that's good for us. Cellulose, why plants use it is that they use it to make these things called cellulose fibers, which are used to create a cell wall. So this is actually a scanning electron micrograph, so a picture taken from an electron microscope of a cell wall of a plant. And plants are unique because they've got these really tightly woven threads of cellulose which form this barrier known as the cell wall. Us animals, we don't actually have a wall like this around our cells, but plants do, and it helps to give them a little bit more protection from the environment. It also gives them a little bit more rigidity, so plants don't have a backbone or they don't have muscles or things like that, but they can still stay upright. They still have a stem that can keep them up, and part of the reason they can stay upright like that is because they've got these fibers that are running in their cells and strengthening their structures. So this is cellulose. Chitin, and then is kind of like the analogous molecule for animals. It's also a structural polymer, and it's made up of a little bit of a different type of molecule. It's not so much just glucose, but it's glucose with one of these little pieces attached to it, and it's got kind of this strange glycosidic, or this strange bond that's between the two sugars. I don't care that you know what the structure of this thing is, but where you find this is it's the thing that is kind of the strengthening component in shells, specifically the kind of the protective covering of crustaceans. So lobsters, crabs, shrimp, crayfish, insect, and then even some of the different insects. So insects aren't crustaceans, but they still have a hard shell around them that protects them. And the stuff that gives it that hard shell is this stuff called chitin. Chitin also can kind of be a little bit fibrous. So this is a picture taken from an electron microscope of, I think this is a crab shell that has been kind of dissolved so that you can see these little chitin fibers. All right, so here's a review question for you. Wondering what elements are carbohydrates made out of? If you could draw an example molecule of a 6-carbon monosaccharide, I got some definition and some examples of the following, and then also 2 examples of carbohydrates that provide quick energy, and 2 examples that do structural support. So pause the video and take a quick second to see if you can come up with some of these. All right, here are the answers. The first one, A, elements that carbohydrates are made out of. Remember, it's CH2O. And it just depends on how many carbons there are. So when you're talking about a sugar, you're going to have a variety of sugars that are one right after the other. And on most of them, they have this exact same shape. They've got an H, an OH, and it doesn't. And depending on how the OH and the H are shaped, it can determine what the sugar is, okay? So like, remember the difference between glucose and galactose is just merely one of these OHs on one side versus the other side. And then on the terminal carbons, you'll have the ones on either end, you'll have one of them will have an aldehyde group, which looks like this, and then the other one will just have our good old-fashioned OH and H, but then it's got one extra bond it can make, so it just throws another H on there. So this is something that would be a sugar. Another example you could draw is a ring structure like this if you wanted to. And these, if that's the case, then you'd have 1, 2, 3, 4, 5, 6 carbons. And then on each of those, then there's just a whole bunch of H's and OH's. And I'm not going to ask you to memorize the particular structures so that you could draw exactly of a certain sugar. That's more like something you do in a biochemistry class. But I would want you to be able to spot a sugar. If I had a bunch of different molecules drawn on an exam, could you pick out which one is the sugar? Okay. Again, it's the one that's going to have the ratio 6, or it's going to be the one that has the ratio 1 to 1. And then it just depends on if there's 5. If there was 5 carbons, it would be C10 or C5H10O5. If there's 6 carbons, it would be C6H12O6. You get the idea. All right. That goes kind of hand in hand with this next question. So we've already kind of tackled both A and B. Then C, give a definition and some examples for each subtype of carbohydrate. The definition for monosaccharide is 1 sugar. The definition for a disaccharide is 2 sugars linked together, and then polysaccharide would be many sugars. Examples of monosaccharides would be glucose is a really great example. We'll talk a whole bunch about that later in the class. Fructose is another one, and then this stuff called galactose. Examples of disaccharides, we've got sucrose, which is table sugar. Maltose is the one that's found in grains, and then also lactose, the one in milk. Polysaccharides, we've got 4 of them. We have starch, glycogen, cellulose, and chitin. Starch and glycogen, these are the ones that provide energy to cells. Cellulose and chitin, these are more for structural support. And then just remember that starch is the one for plants, glycogen is one for animals, cellulose is found in plants, chitin is found in animals. All right, how'd you do? You tracking with me so far? If not, maybe go back and rewatch these until you can get this question down pretty well. All right, moving on then to lipids. Lipids, what the word lipid means is just the fancy word for fatty type stuff. Okay, so when we say lipids, we're talking about this kind of type stuff, butter, all right, or fats or oils or things. There's a common theme in lipids, and they're usually the theme is that they're non-polar polar hydrocarbons. They've got lots of nonpolar molecules that are made up of tons of carbons attached to one another, chock full of hydrogens. Okay, so this is an example of what would be called a fatty acid, and you find lots of these lipid things are made out of these fatty acids. The reason why it's nonpolar, again, is because C's, each one of these little lines represents a sharing of electrons between atoms. Okay, so for example, here carbon is sharing its electrons with this hydrogen, this hydrogen, this hydrogen, and this carbon. They're all sharing electrons to give each other full electron shells, outer electron shells. And carbon shares its electrons equally. So there isn't really going to be any majority of time electrons are spent in one place versus another. The electrons are spent the same amount of time here as they are spent over here. So there's no net charge anywhere. Now over here we do have a little bit of polarity because we've got an oxygen, so this would be a slightly negative pole, this would be a slightly positive pole, this would be a slightly negative pole, and then right here this would be a slightly positive pole, okay, because usually negative hogs electrons, so it keeps the electrons longer than it should, so they spend more time on oxygen, so it's slightly negative. But in relation to this huge honking molecule, this whole big thing, this tiny little bit here is not that big of a deal. Okay, so even though you might have a little bit of a polar part here, the majority of the molecule is nonpolar, so this is going to be nonpolar. And if it's nonpolar, does that mean it's hydrophilic or hydrophobic? The answer would be that it is hydrophobic. Water is not nonpolar. Water is polar. So if you have something that is not polar, it probably isn't going to want to mix with water all that much. There's a couple different types of lipids. We have fats and oils, waxes, phospholipids, and steroids. And we're going to just go through each one on this list and talk a little bit more extensively about each of them. The first are the fats and the oils. And these are going to be made out of two things. One is fatty acids, which looks something like this. Again, it's just a whole bunch of carbons attached to hydrogens, and then the reason why it's called a fatty acid is because on the very last, carbon, on one of the ends, there's a carboxyl group. That's hopefully you recognize this group, a double bond to oxygen, and then a hydroxyl group. And if you remember from our discussion of functional groups, hydroxyl groups are slightly acidic because this H can actually leave and disassociate, and it releases hydrogen ions into the solution. And anytime you're releasing hydrogen ions into solution, you're decreasing the pH, and that's going to be increasing the acidity of something. So we call them fatty acids. Another thing that fats and oils are made out of is this thing called glycerol. So glycerol is kind of like the linker. It links. It's the thing onto which multiple fatty acids can link together. So what happens is on this glycerol molecule, it's a pretty simple molecule, just 3 C's, and then each of the C's has an OH, and then there's just a bunch of H's added on to fill up all of the extra binding spots on the carbon atoms. But in each one of these OHs, the carboxylic group can attach and water can be released, and then these can be then linked together. So this is an example of dehydration synthesis again, because you have two hydroxyls coming together, the OH on one and the H of the other. They meet up, they take off, that means water is made and leaves, and then the two compounds are linked together. Well, glycerol has 3 spots because it's got 3 of these hydroxyls, so what can happen is that glycerol can link up to 3 fatty acids to form this big behemoth, which is called triacylglycerol. Another real common name for these are triglycerides. And so triglycerides are the things that are made out of, that are what butter is made out of, what canola oil is made out of, what vegetable oil is made out of, what olive oil is made out of. So any kind of fat or oil is made out of these things, triglycerides. And fats and oils, these are long-term energy storage molecules. and they store tons and tons and tons of energy because they're really super packed, solid molecules. They're really nice and linear. They don't have a whole bunch of kinks or bends generally in them, and so they pack really tightly together, and it's a good way to like store tons of energy in a small amount of space. And so where carbohydrates are energy molecules that we use, on the order of hours or days, fats and oils are the type of energy molecules that we're going to keep on hand for, a long time. So on my stomach, I got a good chunk of triglycerides sitting on my belly fat, right, that I haven't burned off quite yet. And so that's how animals will store their fats. Fats, when you compare like proteins, carbohydrates, and fats have the most calories per gram out of any of those substances. And that's because of the fact that these are so packed together. All right, there's a way that we can further categorize these fats and oils, and that is to figure out if they're saturated or they're unsaturated. And the kind of the quick and easy way to do it is just if they're saturated, there will be no double bonds. And specifically, this would be carbon-to-carbon double bonds. If it's unsaturated, there will be double bonds, and that is going to be carbon-to-carbon again. Okay. So if we take a look, here's an example of a fatty acid, and when you survey all of these carbons, you don't see any place where there's two lines. Okay, so there's just a single bond between all the carbons. So this would be saturated. Something that's unsaturated would look like this, where you have a double bond between the carbons. Whether something is saturated or unsaturated will give it some different properties. Things that are saturated, so that would be something like this, these all have single bonds, and what that ends up doing for the molecule is it makes it pretty linear. Once you start adding double bonds, it actually is going to put a kink in the molecule, which we'll see in the next slide here. So in this one with the double bond, keep in mind that this isn't actually the shape of how the molecule looks. It's just showing you the atoms, how many atoms there are. The shape in reality of this type of molecule with the double bond would be more like a big, there'd be a big bend in it, because the way that double bonds form, it actually pushes the, either end of the molecule outward to make kind of a bend. So these can get really tightly packed together because they don't have a double bond. So what that means then is that they are more dense, and it also means that when you have a whole bunch of these molecules next to each other, there's these things called van der Waals interactions that occur between the molecules. And recall from, I believe it's chapter 2, that van der Waals interactions are these attractions between molecules just because of there's electrons in each kind of. It's not something that I really want to go into too much detail about, but basically it's just when you have a big long molecule next to another big molecule, they're slightly attracted to each other for this reason, of the van der Waals interactions, and that's all I'll say about that. But if you can imagine, you've got a bunch of these nice, sleek, linear molecules, and you've got a bunch of them that are next to each other and they're attracted to each other. This lends to saturated fats being solid at room temperature. Okay, so an example of a saturated fat would be something like butter, because at room temperature, butter is solid, right? Unsaturated fats then would be an example. These are our oils. So this would be vegetable oil, olive oil, that type of stuff, and that's and that means that they're liquid at room temperature. And again, whether they're solid or liquid, just all depends on how tightly the molecules how closely they can sit next to one another. And if they don't have any double bonds, they're nice and linear, so they are really packed. They're going to be solid at room temperature. But if they're a little bit more loose because of the double bonds are kinking the molecules and they can't gather all that tightly together, then they're not going to be as solid, so then they end up being liquid at room temperature and it's an oil. All right, another category here is the shape of the molecule. Now, most of the time, like I said, you have a double bond formed, and it is going to put a kink in the molecule, so it puts a big bend in it. All right, and this is generally what happens in nature. All right, when you have a double bond, it forms a kink. Now, the food industry a while back realized that they could take oils, so things like, things that were liquid, and they could hydrogenate them, and they could also form double bonds. So that's where we get the name trans fat, right? Because for whatever reason they wanted to be able to take a liquid, maybe because it was cheaper, and if they wanted to have the liquid and make it more of a solid texture, just because whatever they were using required, the consumer wanted it to be a little bit more solid, made the product more desirable. Well, they could make it so that those double bonds were actually in a trans shape, and then it actually made it very much similar to the saturated fats. Remember, we said the saturated fats, these ones without any double bonds, this is going to be things like butter, right? Whereas the ones with the double bonds, these are things like oil. Well, when you make it a trans oil, or you take the fatty acid, you make it so that the shape of the double bond is like this and not like this, then it ends up being very much like a saturated fat in its consistency. But unfortunately, that these, for whatever reason, they tend to lead, it's been suggested that they lead to a higher concentration of a form of cholesterol, which is bad for you. It can lead to plaque formation on arteries and even heart attack. So these trans fats kind of took a bad rap in recent years, and people are kind of making strides to avoid using them. Examples of trans fats, these would be things like shortening or margarine. You've probably heard of those. They're both solid type substances, and they're solid because they started as a liquid and then they changed the double bonds so that it was trans and not cis. So all that to say, the shape of these molecules really determines their properties, namely whether they're liquid or they're solid at room temperature. Another characteristic of fats and oils is whether they're essential or non-essential. Essential means that these are not made in the body. And so we must acquire them in our diets. We have to eat them, okay? We must consume them. Non-essential then means that these are actually made in the body, so it's not really a big deal if we don't get them in our diet because your body can just make them. An example of an essential fat is the omega fats. So you've maybe heard of omega-3, omega-6, omega-9. Well, these are essential fatty acids that we cannot make. We have to get them from places. You can get them from like cod liver oil or different types of fish meat, things like that. And just for your information, you don't have to know this, but the reason why they're called omega-3 or omega-6 or omega-9 fats is because on the fatty acid, this last carbon is called the omega fat, or the omega carbon. Omega means, it's the Greek letter for last. It means last. And so if the last one is the omega, you count up to see how many carbons the first double bond occurs. And so in this case, it's this is the first carbon, second carbon, third carbon. So this would be an omega-3 fatty acid. Just FYI, you don't have to keep that in mind for the exam. All right, next category then are the waxes. Not a whole lot to say here about waxes, other than they're really, really long hydrocarbons. So generally these are made from 2 fatty acids. So up to this point, when we talked about the fats and the oils, we only talked about things being 1 fatty acid long like a triglyceride is going to be 3 fatty acids, and each one is just 1 fatty acid. Each of the fatty acids on that triglyceride molecule are just 1 fatty acid long. But in a wax, there's 2 bound together, so it ends up being really, really, really long. So an example would be here. This is kind of the point where 1 fatty acid, this thing, and another fatty acid, this thing, linked up to form one that's too long. And these are super, super hydrophobic. And the reason is because they're super, super nonpolar, right? We said fatty acids are polar. Like if I go back here and we take a look at one. Here's one right here. We said that these are nonpolar, right? But we also confessed that there was a little spot right here and right here where there's just a slight bit of polar activity. But the abundance of nonpolar molecule in ratio to the polar part of the molecule, it's much more in favor of the nonpolar. So these have a nonpolar characteristic to them. Well, that's even intensified in waxes because we do have a little part of the molecule that's polar, but the ratio of that is now twice as much nonpolar. So they're super, super hydrophobic is another characteristic of them. They're highly saturated, so that makes them solid at room temperature. And because they are so hydrophobic, they're often used in nature for waterproofing. Okay, and we even as humans have found ways to use waxes to waterproof things. But in nature, a couple of good examples would be there's usually a waxy covering on the surface of leaves, and that's good because it keeps water both in the leaf from escape. Basically it just keeps the water that's inside the leaf from escaping. But then as a consequence of that, you can see that some water will bead up on the top of the leaf. Another example would be like this is a picture showing the feather of a bird. And generally on the feathers of birds, they have these kind of glands that secrete wax that lines the feather. And so you can see this drop of water, normally it would kind of spread out over the feather. But because all of those molecules of water don't want to have anything to do with anything on this feather that's coated with wax, they instead just kind of stay with one another. And so it beads up as this nice, pretty looking droplet of water. The last category we're going to talk about for the lipids are the phospholipids, and these are super important for our cell membrane. They make up pretty much most of our cell membrane. So all of our cells in our bodies, they have kind of an outer lining or an outer covering called the cell membrane or the plasma membrane, and it's made up of these things. This is what they look like. They have a molecule of glycerol, okay, which we said has got 3 little attachment spots to it. Remember, glycerol is a pretty simple molecule. There's 3 carbons. Each carbon has a hydroxyl group on it, and then you just add a bunch of hydrogens to fill up the rest of the bonding spots. Well, in a phospholipid, 2 of these binding sites have attached to fatty acids. Okay, so what you're seeing here is that this is the glycerol molecule, and each one of these places is a place where a fatty acid is attached. So you've got a fatty acid here and a fatty acid here. And so what can you tell me about this part of the phospholipid in terms of its polar or nonpolar? This part right here is going to be mostly nonpolar, right, because a good chunk of a fatty acid is nonpolar. So that means it's going to be hydrophilic or hydrophobic? Hydrophobic. But then in addition, on the one last binding site of glycerol, rather than binding to another fatty acid, it actually picks up a phosphate group. So that's what you see right here. In the phosphate group, if we know anything about a phosphate group, you look at all of these oxygens that are sucking electrons from places. So there's going to be a pretty positive charge on the phosphorus, pretty negative here, and in addition, it just simply has one extra pair of one extra electron compared to protons. So it just has not even a slight negative or a slight positive charge. It actually has a real negative charge on it. So when you have that happen, then that thing is super polar, right? Because it's got really strong charge imbalance. So this is going to be polar. which means that it is hydrophilic. And so it's kind of interesting that you would have a molecule that's both hydrophobic and hydrophilic. And that's what lends to its unique properties in our cell membrane. So your cell membrane is made up of a whole bunch of these. Here's kind of an example. So if I was to continue drawing this, let's say there's this big thing. Your cell membrane, the thing that surrounds in each one of your cells, is called a phospholipid bilayer, because it's made out of 2 layers of tons and tons of these little phospholipid molecules. So here's, the first layer, here is the second layer. And if that was the case, then this would be the outside of the cell. And then this would be the inside of the cell. And this would be then the cell membrane thing that surrounds the cell. This is also sometimes called the plasma membrane. Well, what's interesting about this, and vitally important to you and I surviving, is the fact that these phospholipid molecules have both a polar and nonpolar part to them. The reason why this is important is because outside of your cell there's a lot of water molecules. Okay, so I'll just draw some water molecules here. And you and I are mostly about 60 or 70% water. All right, so where is that water? Well, a lot of it's right outside of your cells, and then there's a whole bunch of it inside of your cells. Now, the hydrophilic parts of the phospholipids, so that's these little ball things, right? If we go back here, the phosphate group right there, that's in the little ball part of the molecule. So the hydrophilic parts are going to want to face towards the water molecules, okay? And then that means that the fatty acid tails, they face in like this. But then on this other layer, the phosphate heads are going to also point towards the water, and then the fatty acid tails are going to point towards one another. So you get this nice kind of middle part here, which is hydrophobic. And then everything else is hydrophilic, right? So what that means is there's this hydrophobic barrier between the outside of the cell and the inside of the cell. And it's that hydrophobic barrier that allows the cells in in our bodies to make an inside environment which is different than the outside environment. If there was no barrier here, if there was no hydrophobic barrier, then anything in the inside of your cell could just dissolve, and the water could just pass in and out, and it would be very hard for the inside of your cell to be anything different than the outside of the cell. But our cells, we want to keep them very constant, and in a world where environments are always changing, you don't want the inside of your cell to match the conditions that are outside the cell. Like, for example, if I jumped into a bath of salt water, I wouldn't want all of my cells to suddenly become the same saltiness as the salt water, or that wouldn't be very good. Okay, or if same thing for like sugars or pH or anything like that. Your cells the inside of your cells has to be a pretty constant pH, a pretty constant saltiness, a pretty constant temperature, that type of stuff. So it's good that you can keep the inside of your cell different than the outside of the cell, because outside of the cell is always changing, right? And the reason why you can do that is this hydrophobic region of the plasma membrane, which does not allow water to just freely pass through. And we'll talk at great length about the cell membrane and why it's important later, but for the time being, you do want to know that it's made out of phospholipids, and a phospholipid has a nonpolar and polar region. Okay, the last one here on our list here for lipids is steroids. Steroids are interesting because they have kind of a ring structure to them. So this is a picture of a steroid, and you can see that there's like a ring of a carbon ring formed attached to another carbon ring, so they've got this interesting fused ring structure. A really good example of a steroid is this molecule called cholesterol, and that's actually what you're seeing there in the picture. Cholesterol does a few things. The one is that it stiffens the cell membrane. So, if I go back to this picture where we said the cell membrane looks like this, all of this stuff here, this is just fatty acids. So you've got kind of the thing that surrounds your cell is just kind of like a little bit of fat. And fat isn't all that strong, right? If you try to build a house out of butter, I think you wouldn't have very much luck at all. So the thing that helps our cells have a stronger cell membrane, a stronger covering, is that they're embedded with this hydrophobic substance called cholesterol, which is a little bit more stiff. So it makes so the membrane has a little bit more strength to it. Cholesterol is also a precursor to a bunch of important stuff. So like the hormones testosterone and estradiol, these are the things that lead to the different sex characteristics. So testosterone is responsible for the changes that a male will undergo during puberty, and estradiol is converted into different forms of estrogen, which can also do the same thing in females, responsible for the sex characteristics. Cholesterol is also a precursor to making bile salts. Bile salts are important for helping us digest and absorb fats that we eat. So when you eat those cookies that are made with tons of butter, what helps you absorb that fat and store it in your body is bile salts. Also, cholesterol is one of the things that's needed to make vitamin D, and vitamin D is something that helps your body consume calcium and phosphorus, or helps your body absorb calcium and phosphorus. This is actually made from sunlight when the sun hits your skin. And here's kind of a picture that shows just how important cholesterol is. There's a whole bunch of different stuff that cholesterol can go in to then be modified and make different stuff. So cholesterol can be modified to make this thing called progesterone. Progesterone is a hormone which prepares the uterus for pregnancy. Aldosterone is a hormone that keeps salts in your body so that you don't lose a bunch of salts and with it a bunch of water out of your body. Cortisol is a stress hormone that's released during stressful times to help you utilize energy better. And testosterone we talked about. So you don't have to memorize this figure, but I just point out the fact that cholesterol is important stuff. So you maybe have heard, cholesterol is bad, don't eat cholesterol, all that type of stuff, and that's not true. There's certain forms of cholesterol which maybe have an association with increased risk for heart disease or heart attack, but by and large, you need cholesterol. It's good for lots of stuff. Okay, but in either case, it's a steroid, which means it's got a ring structure to them. All right, here's our review question. What types of elements are lipids made out of? And give a description and a function for each subtype of the lipids below. What are the main components of a triglyceride? Draw and label the basic components of one phospholipid. And what is a phospholipid bilayer? Go ahead and pause the video and see if you can work on these, and then I'll tell you what the answers are. All right, so part A, we're talking about different elements that lipids are made out of, and remember they're hydrocarbons, so it's a whole bunch of C's going on and on and on, bound to a whole bunch of H's. Okay, so definitely C and H is the primarily, so basically carbon and hydrogen are the primary elements that compose lipids. There is going to be on the end of one probably like one hydroxyl group or something like that. So there might be a little bit of oxygen, but for the most part it's carbon and hydrogen. Fats and oils, these things are made out of triglycerides. Or these are triglycerides. Triglycerides are made out of fatty acids bound to this molecule called glycerol. And these are used for energy storage, specifically long-term energy storage. Okay, so if we don't need to get the energy really fast, right away, then it could be stored as a fat or oil versus a carbohydrate. And there's a whole bunch of different types of these. You can have saturated fats. You can have unsaturated fats. If it's a saturated fatty acid that's making the triglyceride, then that's going to be solid at room temperature. If it's an unsaturated one, then it's going to be liquid. So we would call the liquid stuff an oil. Okay. And then you can have cis or trans. That tells you the shape of the molecule based on the geometry around the double bond. Okay. Waxes, these are super long, so it's usually 2 fatty acids bound together. And because of that, these are also very strongly hydrophobic. So because of that, they're good waterproofing agents. Phospholipids, these have, it's basically a glycerol molecule which is bound to 2 fatty acids, and then it's also bound to a phosphate group. And that means that they're going to have some nonpolar parts to them due to the fatty acids, and then they're also going to have a polar component to them due to the phosphate. And these are the things that are found on the which make up the cell membrane. That's why these are really important. Okay, and then lastly, the steroids here. These are fused ring structures. And an example of that is going to be cholesterol. Cholesterol is important because it stiffens the membrane and makes a whole bunch of stuff. The main components of a triglyceride we talked about, that's glycerol and fatty acids. Draw and label the basic components of one phospholipid. If you were to do this, you would draw a little molecule here of glycerol, and then you'd attach some fatty acids to it. Pardon my terrible drawing. Hopefully yours is a lot better than mine. Fatty acids, and then you'd also attach to it a phosphate. Okay, 2 fatty acids, 1 phosphate attached to 1 glycerol. And then a phospholipid bilayer, this is what makes up the cell membrane. It's basically 2 layers of phospholipids in which the fatty acid tails of each layer are facing one another, and the phosphate groups of each layer are facing towards the outside, towards the water. Real quick here, one last just interesting thing about glycerol, as long as we're talking about it. This is kind of just FYI for your interest. You don't have to memorize it for the exam or anything. But glycerol is actually kind of has important roles in our society. The first is that if you take oil, okay, oil looks like this. Again, it's a triglyceride, which means there's 3 fatty acids bound to a molecule of glycerol, right? Well, if you take that triglyceride and you treat it with this stuff called lye, which is the same stuff as sodium hydroxide. Remember, this is a base, one of the most basic things out there, sodium hydroxide. And you heat it up, okay, so you take the oil, you add the lye, you heat it up. It will form these 2 things. So basically what happens is that the sodium will actually this OH here will go in and attach here and disassociate that, and then it will actually make an ion out of each fatty acid. Okay, each one of these things will become an ion with a negative charge. So you can see that that's what's happening here. So we have 3 ions now because we have 3 charged molecules. And then the leftover sodium forms a positive ion, which is next to them. So you've got kind of this interesting thing where you have a molecule that's got that's pretty polar, right? Because you got positive and negative charges there. So this part's polar, and then this part right here is nonpolar. You know, it's kind of weird that you'd find an ionic bond, such as the case here, on something that's a big, long hydrocarbon. Well, at any rate, this gives us some unique properties, and this is, in fact, each one of these little fatty acid salts, this is soap. So when you are taking a shower or a bath or whatnot and you're washing your body with soap, what's happening is that the fatty acid part of the substance is actually attaching to all of the dirt and grime and oils on your skin, whereas this little part here, the place that's polar, is actually attracted to water. So all that to say, it allows water to kind of pull off some of that hydrophobic dirt and grime on your body. So that's how soap has been historically made. Another example is if you were to take glycerol, okay, which again looks like this. You got the 3 carbons, each one bound to a hydroxyl group, and then filled with hydrogens. Okay, well, you get the picture. If you take glycerol and you add some nitric acid, which is what this stuff is, you get this stuff, which is called nitroglycerin. So each one of these, nitrate groups would, or nitrite groups would attach to the hydroxyl groups and form this. So here's each one of those nitrite groups attached to this, which was the glycerol. And what nitroglycerin is this. That stuff is explosive. And nitroglycerin is super unstable. So for whatever reason, when you try and put these 3 nitrite groups onto a glycerol molecule, they just hate each other. These nitrite groups want to get out as soon as they can. And so nitroglycerin is really, really, really fragile stuff. If you even had a bottle of nitroglycerin and just tapped it against the wall, it could just totally explode. And in order to make nitroglycerin a little bit more stable, it was added to toluene, which is a different substance, to make trinitrotoluene, which is the same stuff as dynamite, TNT. So glycerol is important for making soap. It's also important for making dynamite. So interesting stuff there. I think we'll stop here and we'll pick up next time with proteins and nucleic acids.